Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts. And now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 322. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Who seems to be dry.
I am indeed dry. We were spared the worst of Hurricane Helene, which has devastated South and North Carolina and Georgia. And I am just so happy that we didn't get it clobbered, but I'm so sad that my colleagues in the upstate are still digging out. The town of Asheville is gone.
I've been seeing on the news, yeah. Too bad. Also joining us from Ann Arbor, Michigan, Michelle Swanson.
Hello, where it is lovely and just the right amount of rain.
No hurricanes there, right?
No.
That's good. Well, for your edutainment, your education, today we have two very cool papers as usual. And we'll start off with a snippet by Michelle.
Yes, and the title of this paper is the Ticagrelor alters the membrane of Staphylococcus aureus and enhances the activity of vancomycin and daptomycin without eliciting cross-resistance. This is led by a team at the University of Liège in Belgium, and they collaborated with colleagues at the University of Tübingen in Germany, University of Copenhagen in Denmark, and Leiden University Medical Center in the Netherlands. So the first author is Kirsten Leeton, and the senior authors are Lancelotto and Ori. So before I get into the paper, I thought I'd introduce Staph aureus, which is a very common organism. I'm sure most of our listeners have heard of it. In fact, the CDC estimates that 1 in 3 of us carry staph on our skin or in our nose. We can share it then by skin-to-skin contact or on shared towels, for example, at the gymnasium or equipment at the gym. Unfortunately, it's usually harmless. However, it has acquired resistance to methicillin and a number of other antibiotics. So you've probably heard about MRSA, which we have to treat with our kind of top shelf antibiotics, vancomycin. And certain people are at higher risk, athletes that share equipment at a gym, people that live together or work together, for example, in daycare, schools, military personnel, or people who are admitted to the hospital where the burden can be high. And Michael will talk more about that. Also, people who have to use medical devices that are inserted into their body, whether it's like an IV line or a catheter, because then you're breaking that important defense, the skin, and it can gain access. And therefore, people who use injectable drugs are also at risk. So again, typically, it can just cause some swelling, something that maybe looks like a spider bite. But if it gets into other organs, other regions of our body, it can cause pneumonia, bloodstream infections, surgical site infections, sepsis, which can be deadly. So we would like to have antibiotics to treat it, but also preventatives, which Michael will talk about. So here I'm going to share this exciting work that is basically looking at the potential to repurpose a drug that's currently being used to treat people with cardiovascular disease. So it's known to block the aggregation of platelets, which are these small cells that live in our bloodstream and they're quickly recruited to tissue damage sites to form clots and stop bleeding. What's interesting is they've done these very large clinical studies with more than 18,000 patients, comparing head-to-head the Ticagrelor to other aggregation blocking drugs. And what they found, kind of buried in the studies, is that there was reduced incidence of particular infections, seemed to have some anti-inflammatory properties. So a number of groups, including this one, did some direct tests, and sure enough, they found that this drug can inhibit growth and also biofilm formation by staph, including MRSA, also some other gram-positives like Enterococcus faecalis and vancomycin-resistant Enterococcus faecalis, also C. difficile, again, a problematic hospital-acquired pathogen. And they also found that people with prosthetic joints that are on this particular drug for cardiovascular reasons also had reduced incidence of staph. So there were really promising results in the clinical literature and also at the lab bench, indicating that this could be an effective new drug that we could repurpose for treating MRSA infections. But how does this drug work? How is it acting? So what I'd like to highlight is the really systematic approach that they took to identify how this drug was affecting MRSA bacteria. And the tool they used was developed and published in 2007 by a group that developed what they called biosensors for compound discovery. So they took Bacillus subtilis, which is a well-used and understood bacterium in laboratory studies, and they engineered the B. subtilis to express a luciferase reporter. So luciferase will make light when it gets ATP. So you can read it. Light is a good readout. You can quantitate its expression. But they drove the expression of luciferase by a promoter that was known to be activated when a cell was subjected to different types of stress, a DNA stress. They had a separate reporter for RNA stress, a separate one for protein synthesis inhibition, a different one for stress to the cell envelope, and then yet a fifth reporter for affecting a particular component of cell wall biosynthesis. So basically, they could take an unbiased approach and expose these Bacillus subtilis strains that had their different luciferase reporters, expose them to the antibiotic, and ask, do one of the reporters strains light up and others don't, basically, to try to begin to narrow down how this potential antibiotic was working. So they did that experiment, and they also used as positive controls an antibiotic known to affect DNA integrity, so causing DNA breaks, or RNA production, protein synthesis, etc. So by doing that, they discovered that the treatment with this Ticagrelor, in fact, did disrupt cell envelope integrity and also appeared to affect a component of cell wall biosynthesis. And in so doing, it mimicked somewhat two known antibiotics, daptomycin and vancomycin. So that was shown in Figure 1. So they wanted to then independently test a completely different approach to ask whether this Ticagrelor was affecting membrane composition. So they used another set of fluorescent reporters to look at cell morphology and division. And indeed, they found that when they treated the bacteria with this platelet aggregation drug, they found that not only was the cell shape altered, the cells got shorter, thicker, more curved, but they also saw that membrane integrity was disrupted. They saw aggregates accumulating in the cellular membrane, that they lost membrane potential, and the cells also became leaky to a dye, fluorescent dye, that normally can't get across the membrane. So they felt very confident then, using these two complementary tests, that in fact this drug was affecting in some way the cell membrane. And again, these experiments were nicely controlled. They used known antibiotics that affect these pathways and saw similar behaviors. So now that they were convinced that the drug does alter the membrane, they wanted to identify which particular pathways in membrane biogenesis or which particular lipids were affected. So they took some really, again, unbiased, broad approaches, wanted to look at whether the lipid profile was dramatically changed. And they accumulated a lot of data, but nothing that really pointed to a particular pathway. So then the approach they took, which was fruitful, was they just asked, what if we assume that it's affecting a lipid in the outer membrane, let's try to just add excess of a particular lipid molecule and see if that will protect the bacteria from the antibiotic, for potential antibiotic. So they did that experiment, and what they found was by adding excess phosphatidylglycerol or excess cardiolipins or excess diacylglycerol, now the MRSA bacteria were protected from what had been a bactericidal dose of the drug. And that was different from the pattern that they saw with this other well-characterized antibiotic, daptomycin, which was only protected when you added excess phosphatidylglycans to the culture. So therefore, they could conclude, based on these three kind of independent approaches, that this ticagrelor does interact with a number of components of the staph outer membrane. And it provides a clue then where to pursue this line of research. So they didn't identify a particular drug target, but they certainly have got strong evidence that it's affecting the outer membrane, and in particular, interacting with some lipids. So then they finished this up with a more practical set of experiments. They wanted to know whether the ticagrelor could be used to treat infections of MRSA that had become resistant to vancomycin or to daptomycin. So they did a simple set of experiments where they just looked at the minimal inhibitory concentration of daptomycin or vancomycin in the presence or absence. And in particular, they took advantage of some strains that they had that were already resistant to daptomycin. So for example, with these daptomycin-resistant staph, they then added the different doses of the ticagrelor, and they found, in fact, that ticagrelor was still active despite the resistance to daptomycin. So that's really encouraging, because it means if we have a strain in the clinic that is resistant now to daptomycin, we have a new potential therapeutic to use to treat. Likewise, they had, in an independent set of experiments that I didn't describe, they had isolated strains of staph that were resistant to this ticagrelor. And they found that those strains still maintained sensitivity to daptomycin and to vancomycin. So the good news here is that this use of this antibiotic can be saved and applied in cases of infections with MRSA or staph infections that are naturally resistant to daptomycin or vancomycin. They also found that stationary phase MRSA are resistant to vancomycin. That was established. And they're also resistant to the ticagrelor when they're in the stationary phase. But if they used a combination of vancomycin plus ticagrelor, now the stationary phase bacteria are killed. So this is another really exciting clinical observation that will have impact in how it could be used in the clinic. So I wanted to just describe that because it shows an example of how, if we study the literature on particular drugs that are already broadly used for other health problems, such as cardiovascular disease, and look deep into the data, we can get clues that they may have other activities, such as against particular infections, and then using these fluorescence reporters to narrow in on what the potential cellular target is and get a real handle on its mechanism of action, I think is a really promising piece of science that can give us hope against the ever-rising problem of antibiotic resistance.
So, Michelle, one of the exciting things that I think that this paper showed is that this widely used platelet inhibitor,
Yep.
which goes under the branded name of Brilinta. Brilinta is used during the first year after an acute coronary event, and it's used chronically at a concentration well above the MIC at 90 milligrams of Brilinta twice daily for the first year after the ACS event, and then at a dose of 60 milligrams of Brilinta twice daily. And I'm impressed that we haven't seen resistance emerge. Now these are early days,
Right.
and this was the first finding. But because they discovered this drug out of the clinical findings of repurposing, it's unlikely that the microbes are going to become resistant to it. And the other really exciting bit is that this drug, when used in concert with some of the others, principally can attack biofilms. And that's the bactericidal activity that we saw when the community was in maximum stationary phase, like you described.
Right.
And there, I'm not going to say Ticagrelor, I'm going to just call it Brilinta, that's easier for me to pronounce, was able to kill things. So this is early days, but it's really exciting because cardiac patients are extremely susceptible to MRSA infections when they're in the hospital those first few days after their coronary, and their heart is extremely vulnerable. And a MRSA infection or MRSA endocarditis is so deadly for that class of patients. And so by virtue of the fact that they're already on Brilinta, maybe that is what's saving their lives. It's the antimicrobial effect preventing the MRSA from getting that foothold in them. So this is a really compelling piece of work that these authors have provided to not only the microbiology community, but the cardiac community as well.
Yeah, and your point about the biofilms, they did cite a 2022 paper that showed that people that were on this platelet aggregation inhibitor did have reduced incidence of staph infection of prosthetic joints.
Yes.
Consistent with the biofilm inhibition. So
yeah, it's cool.
Michelle, you mentioned that Dapto and Vanco both target the envelope as well, right?
Yeah, vancomycin, particularly the peptidoglycan cell wall layer, and then Dapto, the membrane, it depolarizes the membrane.
And so their observation that Ticagrelor enhances those two when you use them together, it suggests it's hitting a different envelope function, right?
Probably the lipid carriers, my bet, given their lipid experiments.
Cool.
And they did say that another really extensive, unbiased drug screen type report that was published in 23, I believe, reported that this drug, Ticagrelor, altered the stability of proteins that staph uses to modify or flip teichoic acid and lipoic acid, which again are components of its cell wall. So a lot of independent data are kind of pointing at that.
So just when you thought you knew everything about cell wall biosynthesis, folks, here we go, upsetting the apple cart again.
Mm-hmm. I love that you can find drugs right there by just being observant with these clinical data, right?
Yeah, and then taking these fluorescent screens where you can do high throughput screens
so cool.
to tell me, is it protein synthesis, DNA synthesis, RNA, et cetera.
And the other thing about using luciferase is you can actually measure the photonic energy. You can measure the light intensity, and that will give you an indication of how well these drugs are turning things on and off. So it's actually not just qualitative and on and off. It's actually can be quantitative by measuring the number of photons that are hitting a detector.
Yeah, and we've got machines that can read multi-well plates, whether it's 96 well plates or larger. So you can really do this in a high throughput manner, unbiased high throughput manner.
Thank you, Michelle.
You're welcome.
Now we're going to hear about an amazing disinfectant technology from Michael, which is not copper, right?
It's not copper, but it could be. This paper is entitled "The Effects of a Novel Continuous Disinfectant Technology on Methicillin-Resistant Staphylococcus aureus, or MRSA, fungi, aerobic bacteria in 2 separate intensive care units in 2 different states." And it's an experimental design with an observed impact on healthcare associated infections. And this study was authored by the 2 investigative teams at Kentucky and Louisiana, and they are Troche, Lawrence, Carinza, Baumgarten, Lambert, Leger, Berthold, Woolsey, and Burks. Dr. Deborah Burks, who many of you know from her days as COVID spokesman for the Trump administration, is now working for the company that manufactures this remarkable technology. And the paper appeared in the American Journal of Infection Control in August, and it's open access. So today's TWiM is a bit of a busman's holiday for me, in that the manuscript addresses how the disinfection of the built clinical environment can impact the rate of healthcare associated infection acquisition, a topic that I have been investigating for the last 15 years of my career. So this paper is going to be a bit different from our usual TWiM fare, in that we're going to discuss a clinical trial whose principal focus was to evaluate a passive and continuously active antimicrobial technology that will simply debulk the built clinical environment of microbes. The intervention that the authors evaluated was whether the placement of a device capable of photohydrolysis of ambient air would function at a rate sufficient to serve as an augmentation to current standard disinfection and other bundled infection control strategies used by healthcare and in healthcare facilities in order to lower this rate of healthcare associated infections. Their augment, or the addition that they put into the infection control bundle, was the addition of this photohydrolysis system. And principally what it does is it takes the ambient water vapor resident in air, and they ask the question, would a sufficient concentration of germicidal particles be generated? And these germicidal particles are reactive oxygen species, enter the clinical environment through routine air exchanges within the study site, which happens to be the medical intensive care unit. So the twist, if you will, was the intervention would be passive. And while at the same time, it would provide this continuous antimicrobial intervention. No human beings need to take part in this intervention. No cleaning crew, no nurses, no doctors, nobody. It's just the engineers had to install it properly. And then as the air goes through the air handling system, the antimicrobial agents that are generated in situ would come out and do their dirty deed.
But I just want to emphasize they're doing
Please.
this study in the context of 2 active ICU units at
Yes.
2 different hospitals. So
There-
all those people and the patients and all the stated activities are happening.
Everybody's contributing.
Yep.
Now, the reactive oxygen molecules are universally antimicrobial in their ability to inactivate and or kill microbes, whether they be bacteria, fungi, viruses, mycobacteria, or even spores. Thus, the hypothesis that they were testing was whether the resulting reduction to the overall microbial burden in a clinical environment with actual patient would be sufficient to reduce the burden on high-touch surfaces within the units that they were studying. And they would simply measure this by looking for the incidence of healthcare-associated infections, asking the question, what was the infection rate before they introduced the system, and what was the infection rate while they were using the system? So first, I thought I would explain what photohydrolysis is. Well, as its name suggests, it's a process that employs light energy. Here it's ultraviolet light because it has more energy and it's safer than X-rays. The energy profile of the UV light is often fractionated into distinct wavelengths, and there's specific types of bulbs that will generate different types of wavelengths. For example, UVA generates a wavelength between 320 and 400 nanometers. UVA penetrates deep into our skin and it can even pass through glass. Now, UVA is not all that safe as it damages our skin, resulting in its changing of color. We become tan. It's the primary radiation also used in those tanning beds, which dermatologists tell us you should never use. It causes almost all forms of skin aging, including wrinkles. UVB is between 280 and 320 nanometers, and it damages skin cells. And this radiation activity causes DNA mutations that we all learned once upon a time, like thymine dimers, that can eventually lead to melanoma and other types of skin cancer. UVB radiation from the sun can also cause cataracts. And cataracts, it happens when our proteins in our lenses get damaged by this UVB radiation, and they then collect pigments that then serve to cloud our vision. UVC, that wavelength that these folks were using, is between 200 and 280 nanometers. And UVC has the strongest germicidal effect, and it is widely generated from mercury vapor lamps, where the energy packets serve to inactivate microorganisms because of this photohydrolysis effect. It cleaves water into hydrogen as well as free radicals, oxyanions, hydroxyl ion, other things. Recently, though, we have seen the development of light-emitting diodes that can generate UV energy at wavelengths between 266 to 279 nanometers. How the UVC energy kills is multifaceted, but it's principally through the generation of these free radical oxygen grenades. But here in this application, the UVC is literally interacting with the water vapor in ambient air. The process can be harnessed to produce these highly reactive hydroxyl radicals, which are then powerful disinfectants, almost like hydrogen peroxide, but except on steroids. And they can interact and hydrolyze nucleic acids, both DNA and RNA. They can serve to bleach proteins. And within the water of our microbial cells, they can transfer that high energy electron associated with the hydroxyl ion or the oxyanion to water in our cells. Remember, we have a water-based chemistry forming hydrogen peroxide and other singlet molecules in our cells that in turn literally are creating grenades inside the microbial cell that then blow holes in the membrane that result, just like Michelle was talking about, in the collapse of the membrane potential of the microbes. Thus, the microbe is dead at least by four independent mechanisms, which in real time results in a bottleneck where it's extremely challenging. And remember, all selection is at the level of the population and not the individual. So it's very challenging based on how quickly the free radicals are interacting with the population of microbes to genetically engineer or select out from that population. And typically, there's an insufficient number of microbes in that population in order for natural selection to play a role on the time scale of the oxyanions being generated in order to facilitate the development of resistance. Simply put, when the concentration of active agents go up, then the population simply can't keep up and death of the community occurs. And that's the trick. It's the death of the community that is actually going to lower the incidence of healthcare associated infections. It's not that individual microbe, it's the community of microbes that the patient, the healthcare worker will transfer to the patient, or even the visitors, that can actually cause the healthcare infection rate to go up. So how is this going to work in the built clinical environment? Well, the technology that the authors employed builds on the science of photolysis, which we have known for quite some time. But they have cheated and have added a proprietary photocatalyst that is surrounding the bulb that generates the UV light. And this catalyst is a proprietary plastic that has baked into it a metal that effectively increases the activity of the system. And typically these metals are something like zinc aluminum oxides. And I gleaned that from the literature, so that you have a honeycomb shaped photocatalyst made from a hydrophilic polycarbonate material, so it can interact with the water. And then this blend of metallic semiconductors that is proprietary to the company, so they don't define it. But the literature talks about these zinc aluminum oxides. So I'm guessing it's either zinc aluminum oxide or another transition element combination, something like manganese aluminum oxide, where they have enough of the valence electrons available to interact with the photons in order to generate a stronger UV emission spectra, which can then serve to cleave more water vapor. And at least from the literature, the zinc aluminum oxide nanofibers emit two bands peaking at 5.4 electron volts, or at 230 nanometers, and 4.75 electron volts, or 261 nanometers. Bottom lining this is simply amping up, no pun intended, the electrical energy needed in those particles to land somewhere, namely the built environment, splitting the water, energizing the oxygen, or if you will, it serves to enhance the germicidal capacity of the bulb, enabling it to activate more of the airflow, which has always been the problem. How do you deliver enough active agent without running into trouble?
So, Michael, that doesn't sound like cheating. That sounds like innovation.
It is. It is. It is.
Genius.
It is innovation, Michelle. This is brilliant. Now, one of the questions that Michelle tossed over the transom to me earlier when I was pitching this paper to our team is she was worried about, what's this going to do to the patient? What's this going to do to the healthcare worker? And anyone who knows about the effects of ultraviolet light, it can generate ozone. And ozone, of course, is certainly one of the byproducts that can result from photolysis. But I'm here to tell you that this device they had evaluated, and it has cleared through an independent evaluation. It has cleared all the hurdles associated with the FDA safety criteria for a Class 2 medical device, which simply means that it can be used in a patient care setting safely. And again, the concentration of ozone cannot be greater than 0.05 part per million, or 0.107 milligrams per cubic meter of air. From the perspective of the patient in the bed and staff working in the hospital, OSHA, the Occupational Safety and Health Administration, states such devices cannot manufacture more than 0.1 ppmv, or 0.2147 milligrams per cubic meter of air. So ozone is really tightly regulated by all the regulatory agencies because we know it can cause harm. And this technology received the seal of approval from the FDA.
So, now, well, does that mean that while these free radicals can wipe out the bacteria and fungi and viruses, they won't affect the people's microbiome on their skin or the people's cells?
They probably will affect their microbiome to some extent. But again, it's an issue of concentration. I think given the data that they generated, that we're at that sweet spot where we're not harming the patient because they had to conform to the regulatory
Or the
standard.
healthcare workers, right?
Yeah, absolutely. Because you're the ones who are going to be exposed to 365 days a year,
Right.
especially since most healthcare workers don't get vacations.
Yeah.
They're always working. And so, again, as Michelle brings out, the U.S. Food and Drug Administration has a specific class that's the 510K safety clearance evaluation. And they successfully concluded and they were cleared in 2020. So before this entered the clinical trial, they had clearance. So they just didn't put this into an ICU room without already receiving clearance. And that's the wonder of the IRB. They're making certain that patients involved in clinical trials are not put at an adverse risk.
Well, with that reassurance, thank you, Michael. I'm really eager to hear the data.
So they, as mentioned at the outset, the study investigated the effect of photohydrolysis would have on environmental surfaces. So here they used as microbial indicators the concentration of methicillin-resistant Staph aureus. It's a great surrogate for persistent microbes in the built clinical environment, because other studies have shown that Staph aureus can specifically hang around, especially MRSA, in the built clinical environment for months and still retain its ability to cause an infection. They also looked at fungi. And in this era where we are seeing the emergence of Candida aureus, an even greater curse than MRSA in terms of disinfecting a hospital room. And they also looked at aerobic bacteria and fungi in the air. What they did in their study is that they compared the baseline pre-activation sample concentrations of these indicator microbes against the values they witnessed from samples collected once the system was switched on. And again, they used 2 hospitals. They used the ICU in Louisiana, and they used an ICU in Kentucky. And sampling occurred at these 2 hospitals. They have baseline, and it's important that they had 2 hospitals because not only microbial distribution is stochastic in nature, but so are HAIs. You've been to one hospital, you've been to one hospital. You just can't predict because all the patients are different. They're coming in with different comorbidities. So it's a really hard study to do, and they should be commended for the rigor in which they conducted
Yeah, and as an example, they collected samples from the floor, from various surfaces, every 4 weeks for 4 consecutive months.
And they mirrored that so that the time they collected it, if they were in a period of the year where there were greater number of respiratory infections, they made certain that their pre-evaluation period was the same time of year as was after they had switched on the system. And the high touch point surfaces that they used were the ones as defined by the CDC. So bed rails, bed frames, movable lamps, tray tables, etc. And they sampled not 1 square centimeter of surface area, but 100 square centimeters. And so they were following the standard environmental practices of the CDC. One thing that you should be made aware of is that the staff were not blinded to the installation of the system. And that was probably for risk because they had to be told that they were being exposed to this concentration of free radicals. But a lot of people fault the design of the study saying, well, the staff is just going to clean those units better. And I argue always that bacteria don't care if the staff are cleaning better. They'll just come back. Bacteria always find a way. And finally, the one last piece of data you need to appreciate is that they had 1 trained infection preventionist at each facility to assess for the respective HAIs according to the standard practice of reporting.
And again, HAI for our general audience
is healthcare.
Hospital or
Healthcare
associated infection.
And the HAIs were totaled for 21 months prior to the activation. So they went back 21 months, which is a long time, because to call an HAI, you have to spend at least a half hour with that patient's medical record in order to say, yep, they caught that in the hospital. It's a true HAI. So this is a costly study to do because those infection preventionists don't work for free. And then they compared 12 months after, 21 months before and 12 months after activation at the Louisiana and Kentucky sites. Another important bit you need to appreciate is each pre-activation observation period, again, and post-observation, they're mirroring it, covering the seasonality of the respiratory disease months. Now, there's a great map in this open access paper where the rooms were, so you can see how their sampling studies were done. This enables you to quickly walk with them as they sampled, and what did they learn? Well, they learned, like we did with copper back in 2013, that when you augment current infection control protocols with a systematic approach that can passively, and underscore this word, continuously disinfect high-touch surfaces, you're going to witness a sustained reduction in healthcare-associated infections. And indeed, that is what they saw. They observed a 70% reduction, folks. 70. That's a big number. The infections that they were measuring were catheter-associated urinary tract infections, commonly abbreviated CAUTIs, CAUTIs, CLABSIs, or central line associated bloodstream infections, our friend Clostridioides difficile, and the bug we talked about the last snippet, MRSA. They observed a decline in total aggregate counts of HAIs for everything. They looked at CAUTIs, CLABSIs, C. diff, MRSA bacteremia, where the MRSA is recovered from the blood, and MRSA pneumonia, which has an extremely high mortality rate in not one, but both ICUs, with this being observed when compared to the historical count. And
that is amazing.
And they have the most crystal clear figure that anyone could actually look at. You see, at Louisiana, they had 24 HAIs in the pre-period and 7 in the post-period. And they have them color-coded so you can see how many were CAUTIs, how many were CLABSIs, how many were C. diff, and how many were MRSA. Kentucky went from 10 to 3, folks. And they also saw statistically significant reductions in microbial reservoirs. So this is bugs in the room across the environment of care. And this was measured by fungal loads, where the mean fungal CFUs were reduced. And I found this fascinating. 98% of the fungi were reduced in Louisiana. And anyone who lives in Louisiana knows that's no mean trick. I mean, fungi are everywhere in Louisiana. Similarly, Kentucky was 99% from baseline to post-activation. Additionally, they also witnessed a reduction in the mean floor fungal CFUs by 99% at the Louisiana site, 98% in Kentucky. And not too surprising to me was the intervention was also responsible for reductions to the environmental air bacterial, where they're literally sampling air and fungal CFU concentrations in air. They also saw the mean aerobic bacterial loads drop by 72% from 27.3 CFUs to 7.7, and 89% from 10.6 down to 1.18, respectively, from baseline to post-activation at Louisiana.
Yeah, and that dropping of the counts of microbes in the air is consistent with them also seeing a reduction in pneumonia caused by MRSA during this period.
So summing up, this study clearly demonstrates that despite the heroic efforts by the environmental services team, our nurses, our physicians, other staff who interact with patients, serious microbes can remain on surfaces, floors, and in the air. The environmental surface and floor results demonstrated this statistically significant reduction in mean fungi and MRSA CFUs from baseline to final post-activation at both sites. Surface locations with more than 500 colony forming units per 100 centimeters square of MRSA were reduced by 100% at the Kentucky site and 90% at the Louisiana site. So why does that number of 500 matter? Well, between 2.5 and 5 aerobic colony forming units per centimeter squared, so that's 250 to 500 per 100 centimeters squared, are considered the threshold of microbes that are needed to substantially increase the patient's likelihood of acquiring an HAI. For frank pathogens like MRSA, that number is 1 or 100. So again, you see this technology of just squirting a few reactive oxygen grenades into the air was able to do the trick. Now, bed rails, this is the something that the patient is continuously interacting with, shedding onto. Bed rails were the only location where they routinely recovered more than 500 colony forming units per centimeter squared on the bed rail. And this was associated with an active MRSA infection. So the patient is probably just shedding their own microbes onto that site. Bottom lining this, these folks accomplished a lot. They demonstrated that this process of continuous decontamination of environmental air, surfaces, and floors can interrupt that air surface air nexus and subsequent acquisition of microbes that can lead to HAIs, all without the need for additional skilled labor, increased cost of cleaning and disinfection practices, or even supplemental training. Now, if I might, back in 2013, I'm going to take an editorial position here. We published a series of papers where we introduced copper surfaces as a vehicle to continuously and passively reduce the bioburden associated with high-touch surfaces. What we found to be most remarkable, and this is my editorial comment, and we got a lot of pushback from our reviewers. They thought our number, we reduced the infection rate of 60 to 69%. They reduced the rate to 70%. And I found that when you employ a continuously active disinfection strategy, we saw similar results, because I think 69 and 70 are pretty similar. Mm-hmm. We also collected the Apache scores of patients in beds. An Apache score is an estimate of ICU mortality based on a number of laboratory values and patient signs, taking both acute and chronic diseases into account at the time of the patient's admission into the ICU. Here we learned that our intervention was tied to the Apache score, the hypothesis being a higher Apache score at the time of patient's admission into an ICU, which suggests that the patient was more likely not to benefit from the intervention, as they themselves would likely be the source of the infection. So I'm wondering if this team similarly looked at Apache scores. So this technology is going to pay handsome dividends in units other than the ICUs, where the patients are effectively healthier. They're not going to have really horrible Apache scores where the intervention will have no effect. And I think we're going to see HAI reductions greater than the 70% they saw once they move into other units. But there it's harder to do because the patients are ambulatory, moving around, and it becomes a much harder lift.
So speaking of HAI reductions, we were able to contact Kimberly Trosh, who is the first author and director of clinical affairs at the company Active Pure. And she writes, an exciting memory that I have is when all the authors were on a call going through the initial patient outcome results, and we realized we had a 70 and 71% reduction in HAIs at both sites after installation of the disinfection technology. The call got quiet for a moment because that was the first time we realized what we had. The environmental bioburden reductions now had a new and even more impactful meaning because of this replication in patient outcomes and simultaneous reductions in pathogen acquisition.
Yeah, the huge innovation here is the air doesn't have to pass through a unit and then get filtered or sterilized within that unit and then be redistributed, right? They're sending out the active molecules out into the air so it can touch everything, the air we breathe, every surface that is in contact.
And we're not doing anything expensive to the air handling system because in ICUs, an ICU has to have 6 air changes per hour. And 2 of those air changes must be from outside air. Right. And the ambient humidity must be set, and this is defined by the Joint Commission, the accrediting body for all hospitals, must be between 40 and 60%. So there's enough water vapor in routine air that is entering the hospital for when hospital rooms are too dry, below 40% relative humidity. Then we also know from the literature the incidence of respiratory infections increase. So this is already baked into the engineering of hospitals. We just need to put these bulbs in the air ducts.
I think we need to also make sure that nothing happens to the people who are working there, right?
Yes,
well,
yes.
I'm sure they're beginning to do follow-up trials, seeing if their eyes are irritated, because some people may be more sensitive to the free radicals than not.
Yeah.
Tell us more about Kimberly.
So Kimberly is interesting. She has a BS, a Bachelor of Science, and an RN. And so she said her career path began with biomedical engineering. Her dad is a mechanical engineer who really influenced her. And she wanted to help people, and she thought she could do so through medical science. So she went to college at Duquesne. And in her second year, she was a bio major, declared nursing as a second major because she felt that engineering alone wouldn't give her the clinical background that she needed to make a difference. And so that's how she ended up with both degrees. And she wants to thank both the Oxnard Health Center and Lexington VA healthcare system, who have become partners with the company from the initial study design to the manuscript. They supported the research, and they're involved at every step. She also gives credit to her husband of 3 years, Eric, who is not a scientist, but she had him proof the paper and do other things. And she says now he is terrified of MRSA.
Rightly so.
Her advice for aspiring scientists is to have the courage to allow your passions to pave your career path rather than settling for the paths previously made, and always seek collaboration and feedback from peers in your work, because no matter how good you think your work is, there's always something that can make it better. I really like that. There's always something that can make it better, right?
Absolutely.
And that can be other people helping you and give you an advice. So you shouldn't think that you can do everything by yourself.
That is so neat that she's got training both in biomedical engineering and then in the healthcare field as a registered nurse.
Yeah, she wrote that, for example, if I were learning about the mechanical properties of a prosthetic heart valve, I wanted to understand why the patient needed a valve in the first place. Nursing gave me this clinical knowledge. So that's very cool.
She can probably easily envision the lives she's going to help through this technology.
So Michael, what do they do next here with this?
I would increase the sample size. That one of their limitations, they comment that the bioburden didn't get reduced, and they have limited number of samples, so they didn't get statistical significance. They can get that with higher sample size, and because the built environment is stochastic and you have fugitive emissions, people vomit, people lose things in the room. And so consequently, you can have higher rates, and the cleaners don't always catch everything. And so as they increase the N, they can see a difference. But I would If I was the hospital administrator, I would immediately deploy this into the highest risk units. I would put it into medical ICUs. I would put it into surgical ICUs where patients are recovering, and I would also put it
Transplant units.
and I would also put it into the hem-onc unit where you have the immune compromise.
Yep.
The general patient care units, I would investigate, but those are harder studies to do because hospital administrators want to see a reduction in HAIs, and those are expensive studies to do. It takes 30 minutes to review a patient's medical record because we don't have a good algorithm that can call an HAI.
Maybe with chatbot right on that.
Maybe with HAIs. And that was one of the things that they didn't measure in their study. They didn't measure the incidence of ventilator-associated pneumonias. And VAPs are really the judgment call whether the VAP is acquired is an HAI or whether the ventilator-associated pneumonia is acquired from within the patient. And that has a very high kappa statistic where you get a lot of inter-rater. The reliability factor isn't as consistent. And unfortunately, the algorithms and the infection control community and the infectious disease folks always have fights over the new algorithm calling VAPs. You can ask Daniel about that. He'll tell you the great VAP war.
Okay.
And with the pandemic, right? Certainly this technology would also help. Is there any reason to think it wouldn't help with COVID in the air?
No, I don't know.
No, it would. And in fact, back in the bad old days of October of 2020, I reviewed something very similar for the New York subway system.
Yeah, we had on TWiM early in the pandemic, a fellow from Columbia who spent his career working on UVC, talking about putting UVC bulbs in places like train stations. And then when the traffic is low, you turn them on because you don't want them to be on with people there, right? But this is even better.
It's in the ductwork, so you don't have to see the light.
You don't see the light, and it can work continuously without harm. That's really cool. I always wondered when UVC would make an impact on COVID, but yes, it definitely.
I think it's the honeycomb and the zinc aluminum oxide or the manganese aluminum oxide or whichever transition element they're using to create the excitation in that outer electron shell to throw those energy spectra that can split more water. But it's a concentration effect.
Michelle, COVID, influenza, RSV, all of them, right?
Yep.
Yeah, in common areas, this would reduce it. Even sports arenas, right?
Schools, Vincent.
Schools, of course. Anywhere you have a air handler.
Yeah.
I guess you could put it in homes too eventually, right?
Yeah.
That's very cool. Very exciting.
Exciting. All
right. That's TWiM 322 show notes, microbe.tv/TWiM. Questions, comments, TWiM@microbe.tv. If you enjoy these discussions about science, we'd love to have your support. Microbe.tv/contribute. Michelle Swanson's at the University of Michigan. Thank you, Michelle.
My pleasure. Thank you.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.